All right, so let us get going here. Pen. There we are. So today and Friday are actually an experiment. I've never tried to organize the material in this order. And over the last couple of years, I've gotten suggestions from both TAs and people in class about trying to do this. And so I have tried to integrate our discussion of cortex and thalamus. And I hope you'll see why I've tried to do this. It's very hard to talk about one without having the other sort of as a visual aid to bridge it. And we're going to do them both today. Today we will do the basic organization of the two, and we will do the very beginnings of specific components of it. Assuming we have time, which I'm not sure about, I would like to get through at least the visual system as well as hopefully auditory. If we don't make it to auditory, I'll shift it until Friday. We'll do the same thing on Friday for other simple systems, and then next Wednesday we'll come back and talk about some of what we think about as sort of higher cortical function, the things that you do that are not intrinsically linked to one modality, but are in fact much more diffuse processes that require lots of components. So where are we now? Well, we've really made it all the way up to this sort of block here in the middle. And in a sense, we've been aiming for this ever since the beginning. We started off with two tracks in the cord, one of which is going there and one of which is coming from there. We began with corticospinal as our first major descending motor. Well, now we're going to find it, not today, but on Friday, finally back up in the cortex. And we talked about one of the two first sensory tracks, the spinothalamic, and we are finally getting to the destination of that. And that, too, we will not actually get to until Friday, but we're at the thalamus itself. And really what we need to think about for this is the idea that these work as a linked pair and that they sit between everything we've talked about before and each other, so that things come in via the thalamus and to cortex, and from cortex they go out to effectors, but also back to the thalamus, and in fact back to the other structures we've talked about, as everything is highly reciprocally connected. So when we think about them conceptually, we want to think about cortex first, because that's what you can sort of get your hands on in a sort of very straightforward way. It is the point of awareness of input and action for output that is voluntary. Okay? So things that you think about and do, you feel, you see, you hear, you smell, all of that you-ness in that is cortical. Okay? You move, you listen, you turn your head, you respond, you interact, you make decisions. All of that you-ness is the action output from cortex. Okay? So we're going to have information come in, it's going to have to be processed, and we're going to get the output from it. But it's still coming in very discreetly. Okay? It's coming in that my sense of the world is being still put together by what I see, what I touch, what I hear, what I smell, and I don't have separate sort of world views. I have one worldview. So we're going to have to take those distinct modalities, and when they arrive, they will be in what we call unimodal, meaning they are either auditory information, visual information, somatosensory information. We will bring them together in cortical areas that are multimodal, that bring them together and begin to put together world. And then once we have that, we don't just have nothing but a simple reaction. We make decisions. We will assess. We will value. We will figure out the context. Does the context in which you see something make a difference? Yes. Okay? You know, what is the context you're in? What's your memory? What is the prior context that you've seen this in? How do you respond to it? And then you also make decisions. Do you automatically do whatever is placed in front of you? No, hopefully. You are able to make some decisions about deferring, choosing, and the like. And all of that is dealing with information that is not unimodal or even now multimodal, but is really assessing valence and value and making decisions. So all of that is happening in cortex, but it has to begin with the individual unimodal perceptions, okay? So that's the sort of first input point. It goes from there to combined modalities, from there to all of these other types of decisions that you make about it. And then when you're done, what do? Well, you make a decision and you do something, and that's the final output point of your decision comes from the cortex, okay? So you can think of it as essentially an incredibly complex black box. You put in 5 different types of input. It has its own context of prior history, okay? So it's not just running only in the moment. It knows what happened. It has you have memory, you have context, you have the ability to set values and assign valence to decisions. And then you process that, and the only type of output you have is motor output, okay? The other type of output you have, in fact, is attentional. What are you paying attention to, okay? So the best example I can tell you for attention is that right now, as you are all sitting here, until I say the next set of words, none of you is aware of the feeling of your feet on the floor. Now you are all able to be aware of your feeling of your feet on the floor, okay? The sensory input did not change at all, okay? That's all about where you direct your attention, not about input. So you don't get rid of the input. The question is the input being given any attention as you make your decisions, okay? So that's what's happening at the cortical side. So what's going to happen on the thalamic side? Well, some of it is just going to be single modality relay processing. We've gone from a first order temperature sensitive neuron in my foot. Cell bodies where? Dorsal root ganglia, what part of the cord? Lumbosacral, okay? Enters where? Dorsal horn, does what? Crosses the midline or stays ipsilateral? What does it do? Temperature? What crosses? Second order, not that input fiber. So it's got a synapse where? Substantia gelatinosa, crosses the midline, fibers enter what bundle? Anterolateral or spinothalamic. They are from the foot. Where will they end up by the time I come to cervical cord? Pretty far lateral. That whole tract as I come through the medulla, pons, and midbrain will move where? Dorsal or ventral? Dorsal, medial or lateral? Medial or lateral? Lateral. It ends up pretty far out lateral. What will eventually join it? Medial lemniscus, but right now it's sitting out there laterally, and that is going to go on and synapse on the next order neuron in the thalamus, and we'll talk about that on Friday. And then that neuron will project to the cortex. So we go a first order in, second order across the midline, third order in the thalamus, fourth order in the cortex. And we got the information to the cortex. What the cortex does with it, we don't really understand yet. But we've basically sort of moved it forward in that direction. So that we have that. We will have it for vision. We will have it for auditory information. We will have it for taste, which goes with somatosensory information. And we skip it for smell. Okay? So we've got that pathway. That's a one to the next to the next. So how about motor? Why is there a motor component of thalamus? Motor's in the other direction. And we know that it goes corticospinal. It's spinothalamic for sensory, but it's not thalamospinal for motor, right? So motor doesn't sit on the way down. So why does it sit there on the way up? Well, why is thalamus projecting to motor cortex, to motor areas? Well, so what information have we already sent to the thalamus that is related to motor? Yeah, well, not just proprioception. Proprioception is our position sense, but we sent the cerebellar processed information out to the thalamus. And that cerebellar information is about movement and trajectory and position and orientation and velocity, you know, and all of that, okay? So that's information that you need in order to be able to generate those movements. It comes through the thalamus, okay? There will be premotor areas, as we'll talk about on Friday, that allow you to make movements, okay? So we've got those sort of sensory and motor that align with the perception and the action. Okay? But we also have got these components that modulate and drive. Some of them project and modulate multimodal cortical areas, but others are going to modulate. So these are going to be the things that modulate the combined interactions of different sensory modalities. But some is going to be basically, I should have put these in the other order, that sort of general activity level. Because it turns out that network activity, systems that work together, sort of alternate patterns of activity in the cortex. And we know that because cortical activity is what we can measure most easily, either by surface EEG or by fMRI, as we'll talk about later. Okay? So we've got these modulatory components of thalamus, and then we've got the sort of input-output connections of thalamus. So what is the directionality? Almost all of the directionality that we care about is in fact thalamus to cortex. Thalamus as that gatekeeper on the way. But there are corticothalamic projections. We won't talk much about them, but they do exist, and when we talk about output neurons from the cortex, some of them will be going back into the thalamus, okay? But there's also these indirect loops. So if you think about it, we already know one. We haven't just walked all the way through it yet, but we've talked about all the loops, steps in it. So cerebral cortex projects to the basis pontis, right? That's how you make coordination of voluntary movements of your extremities. It's not just the motor neurons projecting to the cord, but you go cortex, the corticopontine fibers. What does basis pontis project to? More specific than cerebellum? Lateral cerebellum and which side? Contralateral cerebellar hemisphere. Okay. What do we know about the contralateral cerebellar hemisphere? What's its outflow structure? Dentate. Where does the dentate project? So two main targets. Contralateral red and contralateral thalamus. And that thalamus goes back to cortex. So we've just made the loop. We haven't previously named all the pieces, but you've got all those parts. To quote Nauta once said in class, you can get from any cell in the brain to any other cell by no more than 4 synapses, if you know your anatomy well enough. Okay? But this is a loop that actually functions and really matters. And we already know that in addition to this part of the loop where we come back through the thalamus, the cerebellum's going to the red is going to the olive is coming back to the cerebellum, okay, but the red is also coming down to the cord, and the cortex is coming down to the cord. And you've got this system now where all of the pieces have to work together in order to give you the ability to take your finger in the air and make that circle, okay? So one of the goals eventually is to try and figure out if somebody can't make that circle, based on what else is wrong, which part of this whole system is what's not working, okay? Because it's not just a simple reflex knee jerk. Okay. So when we think about sensory information, we begin unimodally, and as you'll see in the next block from John Assad and David Corey, the processing of this is very step by step. What you see as vision, as a picture of the world, begins essentially as neurons that respond to typically oriented, moving bars of light that have fixed lengths, or lengths that do not extend beyond a certain length. You know, they are end-stopped. So that's a very rudimentary visual percept, yet you don't see the world that way. You see it as what seems to be a color-rich, three-dimensionally rich surface textured, moving in real-time structure. So you have to go from the simple first neuron up through multiple areas of cortical connections and processing that somehow translate that simplest information into the most complex. And there will be, in general, what we call a first-order sensory cortex that usually is immediately adjacent to multiple other levels of higher order processing. And we just sort of usually go higher order, okay? But then you're going to bring together your vision, your auditory map, and your somatosensory map, and get a world, right? Your world is not made up of separate vision systems, and that brings you to multimodal cortex. And as we'll try and show you, this is going to be sort of in between all of the three major primary cortices. Visual cortex in the posterior portion of the occipital lobe, somatosensory in the anterior portion of the parietal lobe, and auditory in the posterior portion of the superior temporal lobe. And then what's left in between them is the area where you will get to your multimodal cortical things, okay? The motor outputs exist, and they begin in sort of the opposite direction. They begin with a series of planning areas. What is it that you want to do? How are you going to do it? What's the speed? What's the target? How are you going to calculate it all? And eventually, they get from that first initiating area to the final output area, your effector region, and that's what goes down to the cord. So for sensory, you begin at the simplest and go to the most complex. For motor, you begin at the most complex, and then you break it down until you know exactly, I need 6 anterior horn cells that are driving my biceps, immediately followed by 5 anterior horn cells that are going to drive my brachioradialis for me to be able to make this movement, okay? So this is the sort of most simple mechanistic. You can work backwards here very easily. You can work forwards here very easily. The further down this chain you get, the fuzzier and harder it is to understand. And the further back upstream here you get, the harder it is to understand from just looking at electrical activity. Okay? So these two are organized in completely opposite directions. And then finally, the thing that we understand the least is when you take all of your multimodal and you want to try and do all of the assessment of these different parameters. What am I supposed to be doing now? What time is it? What do I usually do at this time? Is this important? Do I answer the phone or do I not answer the phone? You know, you're able to know the difference between answering the phone when it rings, when you're sitting at your desk, you know, at home, just sort of, you know, getting ready to do something, and answering the phone when you're sitting in the middle of an exam, okay? It's the same stimulus. You know to make a difference. How do you do that, okay? It's not about the stimulus. It's about everything that's going to happen in the frontal lobes. And this is essentially what sits in between your multimodal and the beginning of your output. So that you take your input, process it as much as you can, do something with it, and then you go to make an output. And you can make the output independent of context, but under normal circumstances, you will shade it by the context. So when we think about the thalamus, we can separate it out as well. We can think about those that are what we call relay or specific. They're relay. They go between, if you think about the somatosensory system, the second-order neuron sits in the substantia gelatinosa. The third-order neuron sits in the thalamus, and the fourth-order neuron is in the cortex. And in fact, the fourth-order neuron is not just one neuron, but multiple neurons. But you go first to second to third to fourth. It's just handing information off. Now, it is, in fact, processing as it goes, but it's just a relay. It's a stop on the way. Okay? So you have those. You have them for motor function. You have them for sensory function. And as we'll see as we come towards the end of the course, you have them for that limbic, which is where much of the memory and sort of emotional valence comes in. At the same time, you've got the ones that are associational. They are about how do you bring together auditory and visual information. We know that auditory and visual information come together in the brainstem. We use it to do spatial localization. That happens at the colliculus, but that's not about your conscious awareness, about your sense of the world. Okay? So there are those. And then there are ones which are very widespread, and they actually sit in white matter bundles, so they're often known as intralaminar. And these are the least well understood, mostly because they are very hard, relatively hard to study. People are just beginning to do better electrophysiology of them. They turn out to be very widespread in terms of their connections. So if they project everywhere, it's really hard based on where they go to figure out what they do. If something projects directly into the primary visual cortex, you can be pretty sure it has something to do with early stage visual processing. Okay? That's a good bet. If it projects everywhere, you have no clue yet. Yet we now begin to know that these are more about brain state level rather than specific informational transfer. And we'll talk about that on Friday. So what I'd like to do is go through some basic gross nomenclature, and we'll do the cortex first, talk a little bit in cortex about how microscopic architecture varies, think about what that means for organization, and do the very beginning basis of allocating anatomic function to structures. We'll then come back and do a similar thing for the thalamus. The gross is much harder, unfortunately. The cortex is one of the few parts of the brain that you can easily see. It's on the surface, right? The definition of a cortex is gave it when we talked about cerebellum. So it's surface, but why is the basis pontus not a cortex? It's on the surface. So it has to be, what type, is it white matter or gray matter? Gray matter, so it is a surface gray matter structure that is layered. It is organized in layers. Cerebellar cortex has how many layers? 3. Molecular, Purkinje cell, granule cell. What's below the granule cell layer? White matter. What's above the molecular layer? The great empty void of deep space. What's above the molecular layer? What? Yeah, so the P is on the surface, and above it is the subarachnoid space. Okay? It is a surface layered gray matter structure. Cerebral cortex is a surface layered gray matter structure. So it's easy for us to see it and put the names on it. In lab, you can pick the brain up and put your hands right on it. It's easy. Okay? Thalamus, it's deep. It's got fuzzy boundaries. It's got bizarre nomenclature. Nothing is good about it, unfortunately. We'll do some of the nomenclature, and we'll talk a bit about the basic categories of the nuclei. All right. So here we have what hemisphere? Left or right? Yeah, left hemisphere, okay? It is me standing like this over here, facing you, and you're looking at the lateral surface. As you look at the lateral surface, we have color-coded lobes. So we're going to basically divide this into lobes, and then what? Go from a lobe down to the next level of structure, what it's going to be. Well, what do you notice about the surface? Smooth? No. Smooth surface brain for a human is a really bad idea after about 22 weeks of gestation. Okay? Term for a smooth surface brain in a inappropriate context is lissencephaly. L-I-S-S-E-N cephaly. Okay? It's a very bad thing. Lots of interesting mutations are associated with it that I won't talk about. But if you look at the surface, it is folded and it has grooves and bumps. Okay? Why? Yeah. If you imagine the surface area taken up by a sheet of paper, and then you crumple that sheet of paper up, the volume it occupies is much smaller than the volume that sheet held. Okay? It is an efficient way to pack it, is to fold it. And the terminology is that the grooves down are sulci. the singular being sulcus, okay, from the Latin, and the crests are gyri, or gyrus, okay? And so we'll talk about individual gyri within the lobes, okay? When we look from the lateral aspect, we will find one sulcus, we will find one groove here that comes up like this and is known as the, it's not labeled here, but is the Sylvian fissure. Then as we come back on the frontal lobe in the front, we will find our first sulcus that runs all the way up from the convexity down to the base down here. And that is the central sulcus. Everything by definition in front of it is called frontal lobe. Everything behind it and above the Sylvian fissure is parietal lobe until we come to this posterior boundary that is really only seen from the medial surface, where you see a notch. And that's known as the boundary between the parietal lobe in green and the occipital lobe in red. So that is therefore the parieto-occipital notch. On the medial aspect of the brain, there's a sulcus that runs right down frontward that makes it very easy to separate the boundary between the parietal lobe and the occipital lobe. On the lateral surface, we draw an arbitrary straight line down. Then there's another arbitrary line that's drawn. This up here below the Sylvian fissure is the temporal lobe, but at the posterior boundary of the temporal lobe, we lose any anatomic lines, and so we draw another line that runs roughly perpendicular, that is the boundary between parietal, temporal, and occipital. The first consequence of this is that when I, as a neuropathologist, get a specimen from the OR, from exactly the same location, it can be labeled temporal lobe, parieto-occipital lobe, and it all means that it comes from someplace in this vague area here that doesn't really have a good landmark. And sometimes it gets confusing. Were they biopsying two different targets, or is this just, well, let's call it occipital this time. On the medial aspect, those boundaries are easier. So now we've got, yep.
I don't quite understand. Why is it important to have the folds as opposed to filling in the spaces to have, I guess, even more gray matter?
So gray matter it's a layered sheet, okay? So you've got, structurally, you have a sheet of tissue. How do you pack a sheet of tissue effectively into as small a space as you can in order to put it inside your skull so that your head isn't so big that you need muscles like an elephant to hold your neck up?
But you couldn't possibly just scratch it so close together that you basically get rid of them?
So the problem is that if you fold it that way, there are malformations in which instead of having a cortical sheet that runs like this, so it's got a surface layer and then a white matter layer, and has its laminations within it and its neurons. And then you take this normally, and what you do is you fold it like this, and this, and this, to basically decrease the volume, so that this amount of surface area would have previously gone that full length, right? So if you do that, if you then begin to fuse these, what happens is that you end up, first of all, not having the ability to deliver vasculature. Because remember, the vasculature tracks in the subarachnoid space that comes down and up. So you've made tissue further away from vasculature, which is a bad thing. But in the malformations, of which the form is known as polymicrogyria, you also, because the developmental connections happen when it's a flat sheet, your connections are all mostly running this way and sort of this way in that pseudoplane. Nothing is running in a cross bridging like that, and so you end up with funny connections, and so it's very non-functional. So we have frontal, parietal, temporal, and occipital, and we flip to the medial. We can see that the frontal lobe has a medial surface, the parietal lobe has a medial surface, the occipital lobe has a medial surface, The temporal lobe has an inferomedial surface, but there's this purple structure that's wrapped right around the core. Okay? So what has been cut here? Where have I cut? What's that transection plane right there? Well, let's just look. What was this running right here? Aqueduct. So if cut across the aqueduct, I have cut across the midbrain. So I took the whole posterior fossa contents, the whole of the brainstem cerebellum out of the way so I could see. And then where else have I cut? Well, where's the right hemisphere? It's in some other figure, right? Okay? So I've cut right down the midline. So I'm looking at the true medial surface, and I can see that there is this separate structure. There's this gyrus that wraps around the medial surface. It folds in this plane too, and it is completely contiguous all the way around the medial surface. And this is what we call the limbic lobe. This is the cingulate gyrus, as we'll talk about later, coming around to the parahippocampal. But they are a contiguous structure. They are on the edge, and the word limbic comes from the French, where it means hem, and so it is in fact the edge. It's that sort of hem at the bottom of a garment. It's the hem of the cortex. All right, so now let's look. What are we looking at? What part of cortex? What lobe? Colors have changed. We're using the same colors. It's a limited palette, okay? But previously, that purplish frontal lobe is now here recolored. The brain has been tipped. Why has it been tipped? What's its angle? So it's not just a straight lateral view, right? It's been pivoted over so that you can actually see all the way from the central sulcus here, the first one that comes all the way down, okay? You can see that there are lots of areas, and you can see all the way, this is the very frontal tip. This is known as the frontal pole. Okay? And if you look, you can see that, and you'll see in the brains in the lab, that most people have got 3 gyri. They are convoluted still, but they are basically 1, 2, and 3 that run from front to back. But they don't go back to the central sulcus. They actually stop at the sulcus in front of the central sulcus. And what would we call that one? The precentral sulcus. Isn't that great? And what will we call the gyrus in between them? It's the precentral gyrus. So we've got superior, middle, and inferior frontal gyri that are oriented anteroposteriorly. And then we have a central sulcus, central gyrus rather, that's oriented from the convexity downwards. Okay? So we've got sort of, if I schematize it, we've got 1, 2, 3, and a down. Got it.
So are each of these gyri basically their own little things that we folded up?
So they are completely contiguous with each other.
Okay.
So if you go down this sulcus here, you go from this into that. Okay? This is purely about being able to say where you are. Okay? When you flip to the medial surface, you can see that the superior frontal gyrus wraps around the medial surface. On the orbital surface, we'll get a better look at it below, but there's the gyrus right underneath. What's this structure? Okay? This is a reading test, not a memory test. This is an olfactory bulb. It has that thin little straight line. That's known as the gyrus rectus. It's straight. And then the rest are known as orbitofrontal. They sit above the orbit. Okay? And then we have that medial portion that hangs over the midline of the precentral gyrus, which is sometimes called the anterior paracentral lobule. Okay. So we've got that structure to frontal lobe.
When we think about the gyri, do you think of them as like almost like the intestines where it's all connected in one tube, or is it more like a sheet of paper? Right, so is it the sheet of paper?
So they are completely contiguous. If you flatten this out, you would get one huge sheet of cortex for that whole hemisphere. There are no boundaries between them. We'll come to the connectedness. So it all depends on connections. Both where you are, your identity is driven by both where you are and who you're wired to. Where are we now? Which lobe? Occipital. So if you look on the posterior surface, you can see the parieto-occipital sulcus coming from that parieto-occipital notch. If you grab the brains in lab, you will see the parieto-occipital notch. You'll be able to see that sulcus. And here, on the lateral aspect, we're really vague. We just call them lateral occipital gyri. That's really convenient. On the medial, we will have one that we call cuneus, okay, lingual, and then occipitotemporal. It bridges from the occipital lobe towards the temporal. What do you think it's going to get called right over here? Anybody who grew up locally? Okay. So if you're driving on a street that runs from, you know, Waltham into Weston, what's it called? It's called Weston Street, right? And you come into Weston, and what does its name become? Waltham Street, right? That's, you know, there's Newton Street in Brookline and Brookline Street in Newton. They are the streets that connect across them. So as we cross the boundary here, what's this gyrus going to likely get called? Temporoccipital. Okay? All it's telling you is where it is and what it's next to. Okay? So, we've got those. Those are easy. Now where are we? Parietal lobe. So parietal lobe's pretty easy on the medial surface. Here's that parieto-occipital sulcus. What was this called over here? Cuneus. So we get a precuneus. Now it turns out that the precuneus is a whole lot more important than the cuneus, but it's stuck being named based on its neighbor. Nothing I can do about it. What's this on the in green up here? It's called the posterior sagittal lobule or paracentral lobule. This is the central sulcus, and this was the precentral gyrus. What's this? Postcentral gyrus, and this is just it coming around onto the medial surface. And here it is, the postcentral gyrus. Okay? And then we will have what we call superior parietal lobule and inferior parietal lobule, which consists of these two structures. So together, the supramarginal and then the angular are the inferior parietal lobule. So we have an inferior and a superior. The superior usually doesn't get subdivided, but the inferior often does. And now, we had frontal, we had occipital, we had parietal, we have temporal. And temporal is organized from the lateral aspect very similar to the frontal. You've got three: superior, middle, and inferior. They run really quite anteroposteriorly. Okay? And then if you come to the medial surface, you can see that the inferior temporal gyrus wraps around. Here we've got that temporo-occipital or occipitotemporal. It depends on the book, which way you call it. Okay? And then you come up around, and even the superior temporal sneaks in a little bit around the midline. And then you've got this thing that's called the lingula. It looks sort of like a tongue sticking out at you. Okay? All right. And then the portion that wraps right around the middle is the cingulate gyrus. The sulcus that sets it apart on the outside is the cingulate sulcus. Here is the corpus callosum that we will talk about next Wednesday when we discuss white matter bundles. All right? It wraps around, and at this point here, it changes name, even though it is completely contiguous. And that is known as the isthmus. It's the narrow part comparable to the isthmus of the thyroid, the bridge from left to right lobes. Okay? And then from there, we come to what we call parahippocampal gyrus, because the hippocampus, as we'll see in 2 weeks, is deep to that structure. Huh? John?
It seemed like on the slide there were two lingual gyrus, like one in like the temporal. And
That's the continuation of it. Yep.
okay.
That's a little piece of it that's in the other lobe. All right. So, but if we think about it, let's come back to this lateral view. Here's my temporal lobe, and here's my frontal and parietal lobe. What's in here? How do they connect? Okay, you can't see from the surface. Okay, so in fact, do they connect with just a simple bridge? And the answer is no. They connect actually with complete sheets of cortex. So this is back to Tina's question. Here we've pulled open that Sylvian fissure and actually cut away the inferior portion of the frontal. Okay? If this is central sulcus right here. Here we've cut away some frontal lobe. Here we've cut away some, what? Parietal lobe. And you can see that from the temporal lobe, you go in and there's this huge sheet of cortex here that then connects back up. And the cortex that's down there is what's known as the insula. It's also known as the islands of Ryle. This is Ryle. And that's part of that contiguous sheet. And as we look at cross-sections, we'll be able to see it. And it turns out to be very important for autonomic system regulation as well as sensory integration. So we've got these areas. Are they all the same? I mean, do you think this is going to be built? I mean, we know in cerebellum that if you look at it microscopically, every area of cerebellar cortex looks identical. It has Purkinje cells, it's got granule cells, it's got basket cells, it's got climbing fibers, it's got mossy fibers, it's got parallel axons. It is completely synaptically modular. It is only functionally different because of inputs. So is cerebral cortex going to be that way? Just show of hands, say yes. Yeah, life would be simple for you if it were. Unfortunately, cerebral cortex is not built that way. It is, in fact, structurally different by functional domain. And so its complexity and in fact, its repertoire is far greater than your cerebellar repertoire, because not only is it driven by the types of inputs, but it's driven by what its wiring pattern is intrinsically. So, yes, the answer is different areas look different and are differently organized. So it's not just that they are in different places, they are intrinsically different. So when you look, you have a wide range of cells, some of which are very local and project only within small zones, some of which have dendritic fields that are very wide, some of which have very small, relatively basally placed nuclei with huge apical dendrites and exiting axons, and some have axons that run only within cortex itself. So, if we think about it, there's a basic structure that we can go through. So this is a 6-layered cortex. If you go from the outside to the inside, it's the way you name it. And the first layer is layer 1, and it's a molecular layer. As with the cerebellum, it means it's got relatively few neurons. Okay? Mostly dendrites and synapses running through it, and axons running through it. We then come to a external granule cell layer. What does granule mean? Small. Yeah, they're little round cells. Then you come to an external pyramidal cell layer. Those typically are bigger, and you, instead of having a roundish nucleus, you've got a sort of triangular nucleus on a section. All right? You have an external granule, you have an internal granule, you have an external granule and an external pyramidal. What's going to come next, do you think? Internal granule, and predictably then comes internal pyramidal. God, it almost makes sense. And what's left? A little bit of everything. Okay? So here's the structure. Molecular, granule, pyramidal, mixed. All right? So, terminology, because we use it when we describe things. Supragranular is above layer 4. What's layer 4? Internal granule. Everything is referenced relative to the internal granule. So, 1, 2, and 3 are supra. Infra is 5 and 6. And then the term homotypic gets used, which means you see all 6 layers. If it is different from normal, then it's heterotypic. Okay? And heterotypic can be skewed in either of 2 directions. You could have more granule cells and fewer pyramidal cells, or you could skew the other way and have more pyramidal cells and fewer granule cells. And so if you've got more granule cells, you call it granular cortex. And if you have more pyramidal cells, intrinsically, by definition, then you must have fewer granule cells, and so you call it agranular. Why is it not called pyramidal cortex? Beats me.
For the supragranular and infragranular layers, what if you're talking about the fourth layer? Which one is it?
It's neither. It's the granule cell layer. Everything is for reference to it. So what do these look like? Well, here they are. This is layer 1 without many neurons, layer 2 with granules, 3 with pyramidals, 4 with granules, 5 with pyramidals, 6 has got a mixture. Here, we've got fewer external granules and about the same number of internal. We got more. The pyramidal cells are missing here in 3. That's why you can't tell the difference between 2 and 3 much. And you've got a very small pyramidal layer in 5. This is heterotypic granular. These tend to be sensory cortices, usually primary sensory cortices. And here you've got huge numbers, including these are probably Bet cells. Bet cells are the cells that give, these are the upper motor neurons that give rise to the corticospinal tract, and this is what you see in motor cortex. Now, why is this thought to be the norm? Well, why would you define something as being sort of normal and everything else as being sort of different? If you've got, it's by far the most common. How come? Well, how much of your brain you really allocate to sort of first-order sensory processing? Not much, in fact. In fact, those cortices are pretty small. How much do you devote to your sort of final motor output? Not much. Most of your brain is either multimodal or decision-making. And in fact, when people first started looking, most of the samples they looked at were this. They were association cortices, and associational cortices are the vast majority. So these, when they finally encountered them, were thought to be outliers. All right. Yeah, they are functional together. So in fact, the major input, the 5 and 6 are commonly your output structures. 4 and then supragranular are commonly your input, and 4 is particularly your input from motor and for motor and in here and here. It's the input for motor and sensory cortex for thalamus. So the differences in development are, in fact, relatively late. They're not at the pre-migratory or early migratory state, but once things are in place. So early on in cortex, they all look the same. Pyramidal. What will be a pyramidal neuron doesn't look any different. There are different transcription factors, in fact, that mark them, and that are sort of identity defining, that we actually can use for pathologic things where we think there are the wrong type of cell in the wrong place or a correct type of cell in the wrong place. Those are cortical malformations, cortical dysplasias. Chris Walsh will be here in December, and we'll talk about migratory abnormalities and dysplasias and the like. So just other cortical patterns. The cerebellum is 3, and we'll talk about the olfactory structures and hippocampus later. So if we've got different patterns, there are different patterns, we think we can tell something about what function might be from the pattern, right? So how are you going to organize this? Well, wouldn't it be great if all of superior and middle frontal gyri were homotypic cortex, and the inferior frontal gyrus was granular cortex, and the precentral gyrus was agranular cortex, and the postcentral gyrus was granular, and then superior parietal lobe was homotypic. I mean, it would be great if they were just lined up with gyri, but they don't. So how are you going to try and organize this type of information? You got to build a map, okay? You have to have some map of what type of cortex is where, and then onto that you'll be able to overlay function and connectedness and see if you can understand how this is going to work. All right? So in fact, the maps we use are the maps from Brodmann, okay? And the maps we use are more than 100 years old, and they are still the most useful map that we have, by definition. I mean, there are lots of subsequent versions, but this is the map. And it's complicated, but I'll tell you it's nowhere near as complicated as more recent maps that people have made, but they are absolutely useless because they are so complicated. Whereas this is one that is actually, it's manageable for those of us who do neuroscience, okay? And you will see that areas are named as Brodmann area. So you will read papers and they will talk about primary visual cortex and they will specify it as Brodmann area 17. And this is Brodmann area 17. So where is that? Lobe? Occipital, medial or lateral? Medial. And this, in fact, turns out to be what we call the calcarine sulcus, and this is the calcarine cortex on either side of it. What is going to be immediately adjacent to it here in Brodmann area 18? This is going to be first-order visual processing. That's going to be the beginning of second-order processing, immediately adjacent. Okay? And as we go, I will talk about distinct Brodmann areas. You will not need to know all of them, but the ones that you need to know to be fluent and understand where lesions are and where functions are, we will emphasize as we go. So this is the map that we will be using. Notice the anatomic boundaries that we define for lobes typically do not align in any meaningful way with Brodmann's map. The major exception being pre- and post-central gyri, with pre-central being Brodmann area 4 and post-central being Brodmann area what we call 312. Okay. Here's just a relatively recent, it's a couple of years old now, from Davy Van Essen. This is their map. It's a beautiful paper. It's got wonderful, incredible, high-res functional and structural MR data, and no one uses it. Absolutely no one. So when we think about these areas, how can that map be meaningful? If you look at the you've got two hemispheres and a third brain in lab. If you look at them, they are different, right? The patterns of folding are different, okay? Everybody will have superior, middle, and inferior frontal gyri, but they will be convoluted in different ways. How are these structures consistent from individual to individual? So what's being done here is, in fact, taking fresh human brain and dissecting the cortex off and flattening it out, okay? And so if you do that flattening it out, you can actually say, now if I did my histology on it, where would the boundaries be? What does the structure that I've drawn on that surface map look like? Okay? And in fact, this is done here for the primary visual cortex in a fresh human specimen. And basically, you have this rough oval that's oriented like this. And by the end of the morning, we will be able to put the world onto that oval. Okay? But that's this brain. How about the next brain? So this is actually an elegant paper that was done both in humans and in macaques. Jean is actually out sick today, so don't let me forget at the end. Those are in her group. We're going to have to redistribute you because she's not here. But Jean, along with others at the Martino Center, went and basically by high-res imaging were able to see individual structures and identify area 17, the primary visual cortex, in multiple people. And basically showed that if you look and you take these convoluted structures and inflate the brain so you've made it into a flat surface, everybody's is in fact a consistent oval with a very narrow range of major and minor axes differences across people. So in fact, these are very well preserved anatomic regions, even though the crumple of the paper is very different person to person. Okay? So that there is enormous consistency in cortical organization, even though the cortical surface folding is not. So, in general, homotypic is association. That's why there's so much more of it. Heterotypic, sensory is more granular, and motor has fewer granular, is agranular with more pyramidal. So, the major modalities that we need to think about for first-order sensory and second-order. So, highlighted in dark green here is the motor. That is our motor output cortex, source of the corticospinal tracts. In lighter greens in front of it are things that we think about that are in front, premotor. They are functionally premotor, so we often talk about premotor. You'll see the terms you will see will be premotor, supplemental motor area, and motor planning areas. Remember, that's how you get and give your final instructions that say 73 motor neurons here, 2 motor neurons there in order to make your movement. Okay? Within them, what have we got? Your eyes are really important, not just to me, but to your brain. You have very specific areas for moving your eyes, the frontal eye fields. That's how you decide where to look. I've got an image in front of me. I'm going to make a saccade. My eye does the move. That's those frontal eye fields do the directing for it. Okay. Then, as we come across the central sulcus, we come into the primary somatosensory cortices. Okay. So look, we've got motor and sensory immediately next to each other, but does information really flow from the sensory to the motor? No, because this is the crudest, simplest, least decoded sensory. So we go into somatosensory association areas. Okay? Visual information, as I've said, comes into the Brodmann area 17 in the medial occipital lobe and goes into visual association areas. What's sitting in here that's not colored is the multimodal. What lobe is that in? It's going to be in parietal and posterior temporal. Okay? Then what's going on all up here? All of the decision making. And this gets termed, the horrible term, prefrontal. Okay? Prefrontal should be like the brain of the person sitting in the row in front of you. Okay? You said the whole thing in the front is the frontal lobe. How can you have something in front of it? What it really means is in front of those other designated functions, which are the motor components. So once you come in front of these Brodmann area 6, you come out here into Brodmann area 8, and now you're in what we call prefrontal. And this, what would you name it if you wanted to give it an anatomic zone? Where is it? Yeah, it's dorsal and lateral. So that's dorsal lateral prefrontal cortex. Okay, a very common abbreviation. So you can see we've got primary motor, primary sensory, primary visual, and actually tucked in here on the superior surface of the posterior temporal gyrus is the primary auditory. The sensory, visual, and auditory all surround that parietal lobe where you're going to bring it all together. The information from there is then going to go to the front, to the very, very front, work its way back to the motor planning area, and out comes your output through the motor output. And now you've brought information in, made assessments, and gone to an output. So when we think about it, we can say we've got unimodal association. Those are the light greens. So you go from a first order sensory, a unimodal sensory, multimodal. First order visual to a second order visual to a multimodal. And that comes around on the other side as well. So this is also second order visual. And the same thing happens for auditory here. You go to second order and into the multimodal. And then up front, here it's called multimodal association, but it's really much more what we would consider to be prefrontal and valent. So what I like to think about is what's the meaning and content of those actions and inputs. All right. Let's stop at this point, pick up again at 20 of, and talk about thalamus and try and do the mirror connections between the organization of cortex and the organization of thalamus. So we're going to switch now and work through a sort of similar conceptual structure for thalamus. And as we do that, we now have a set of targets to be connecting it to. And so we'll talk a lot about how things are aligned physically based on the targets that they're going to reach. Right? So the thalamus is actually part of a whole set of structures known as the diencephalon. So we haven't used many of these terms before. The other ones that you might have heard, and I use every now and then, is the term for the midbrain, which is the mesencephalon. Okay? Not commonly used, but you will see it, especially in developmental context, is the mesencephalon. And when we talk about diencephalon, it actually is made up of 4 separate structures. Something called the dorsal thalamus, which is what we mean when we say thalamus. There are other components, hypothalamus, that sit inferior to it, epithalamus, that sits sort of around it, and the ventral thalamus, which, although it is contiguous with the thalamus, we will talk about as part of its functional complex, which is basal ganglia. So all 3 of these we will come to later on. So, dorsal thalamus, that final last contact point, relay point before we go to cortex, regulatory point for motor activity, and modulatory inputs into cortex. So where is this? Well, it's that green lump that in this beautiful image is able to be suspended smack in the middle of the air, but it isn't in the air. It's in fact completely contiguous with adjacent structures. And that's one of the things that makes seeing it such a mess. Okay? So we're going to look at sections of brain, slabs and cross sections, in order to be able to see where it is. Same way we had to look to see the anterior horn, same way we had to see the medial lemniscus. You can't see it unless you look at a cross section. Okay? So where does it sit though? It is dorsal to the midbrain. As you ascend from the midbrain, you go from midbrain into diencephalon and into that thalamus. What fiber bundle do we already know that's doing that? Well, I can name 3 of them already. So spinothalamic has to be doing that. If you have the spinothalamic, what else do you have to have? You have to have the medial lemniscus, because that's the other part of that sensory information from your cord, right? And then what else do we know about that's going that way? The contralateral cerebellum. So it's that those superior cerebellar peduncle fibers cross the midline in the low midbrain, wrapped the red nucleus, and are traveling right on up to the next stop, which is immediately above it, which is the thalamus. Okay? So when we think about where that sits, it is immediately dorsal or posterior or superior, depending on how your animal is oriented. Here is your collicular plate. Okay? Remember, what's happened down here in the inferior colliculus? What have we been doing a lot of? Auditory processing. We will see that the connections from the inferior colliculus come right up like this to reach the inferior boundary on the medial aspect of the thalamus. Okay? That's where its connections are. All those other fiber bundles are going to traverse and run like this. Where's visual information in this picture? So where is it? What's the structure where you can see some visual information? Superior colliculus, but remember, that was a distinct path. That wasn't the sort of visual awareness path. So where is the visual information here? Well, it's still at this point. Optic nerve and optic chiasm. You can't see the optic tract. Why can't you see the optic tract? Where's the cut? The midline. The midline, the optic tracts aren't there. The optic tracts run lateral. Okay, so you won't see them. But that optic tract is running right around the brainstem and is also going to come back to the aspect of the thalamus. And because it comes around laterally, it's going to come out to the lateral bump back here, and that's the lateral geniculate, as we'll see. Everything else is sort of an internal fiber bundle. So now we've got brain and we're cutting slices. So what kind of stain is this? What's black? White matter. Okay? Makes sense. All right? So we're looking at a myelin stain where the white matter bundles are black, and we're going to be looking at these structures in here that are the thalamus. Okay? And the thalamus is basically all of this lump in here. So that's a nice big lump. And as you look at it, you go, boy, it's really hard to tell where I am. It sort of all looks the same, doesn't it? If somebody hadn't labeled it and drawn those nice red lines in for you, you wouldn't know that they had substructure. That's the way the thalamus is. You have to know roughly where you should be. You cannot tell exactly by putting your finger on it exactly which nucleus you're in, because they are not bounded the same way that the inferior olive looks different from the hypoglossal and looks different from the ambiguous, okay? And you can tell if I put an arrow on something, you can tell me whether it's in the principal inferior olive, the dorsal accessory olive, or the medial accessory olive. That's very easy. In the thalamus, that just doesn't happen, okay? You can tell if you put an electrode in and you examine and listen to the types of recordings, because the patterns of activity will be different to different areas. So when we think about them, we will think about anterior structures, lateral structures, and medial structures, okay? We will also have reticular nuclei, meaning that they are embedded within the white matter bundles. They're hard to see on this type of stain. Okay? This is the basic organization. This here would be the, what? Lateral ventricle. Coming through here is the foramen of Monroe, and that makes this space right here third ventricle. So now we're cutting in what plane? Those were actually horizontal cuts, but what are we cutting in now? So somebody? We're going to cut anterior to posterior in the coronal plane. So we're going to slice going forward. So right here, if this is the optic tract, are we in front or behind the chiasm? We're immediately behind the chiasm. And when we do that, we can see here the lateral ventricle, the foramen of Monroe coming into the third. All right? So this bulge that sits right there is what is known as the anterior nucleus of the thalamus. It sits right at the foramen of Monroe. Okay? When the neurosurgeons use an endoscope and pass it from the lateral ventricle through the foramen of Monroe, the two things that they have to worry about damaging are what are on either side of it. The fornix, which as we'll see in about a week, is the major output structure from the hippocampus, and the anterior nucleus of the thalamus on the other side. Okay? So there is our anterior nucleus of the thalamus. And then we've got a nucleus here that comes in, VA, ventral, anterior. So here's the real problem in thalamic nomenclature. It's really hard to know whether something is considered ventral or not. Because if you looked at that, you'd say, you know, if I had to name that, it goes almost all the way up to the dorsal surface of the thalamus, right? Okay? It, you know, the top end of it, where's my pointer? The top end of that here is not that much more ventral than the top end of the anterior. I can see why the anterior is here. It's going to vanish, and that's my anterior most lump. But why is this called ventral anterior? Why isn't it just called anterolateral? Because it's pretty anterior and it's sort of lateral. But it isn't. It's called ventral anterior. You just got to know these, and you can't look at it and go, where is it, what should it be? So now we have stepped posteriorly. So first, watch what's happening. Look, watch the optic tract down there at the bottom. Which way did it move? It goes like this. Moves lateral. Why is it going lateral? What does it have to do? It's going around the brainstem. Okay? You can't go straight back. The brainstem's in the way. It's going around the brainstem. Okay? So now we are behind. We are further back. We've got a little bit of anterior nucleus. And now instead of having ventral anterior, now we've got something that's called ventral lateral. Well, is it more lateral than the ventral anterior was? Not really. You know, I'm sorry. They're not my names. They're just what they are. But we will see at this level we've got the anterior nucleus, the ventral lateral, and now this one. This is called dorsomedial. But 50% of the ventral lateral is more dorsal than any of the dorsomedial. All right? You just got to remember these things. Unfortunately, you can't use the anatomy. So we've gone from seeing anterior and ventral anterior to seeing anterior, dorsomedial, and ventrolateral. And as I'll show you in a bit, the ventral anterior and the ventrolateral are going to function as a unit, and they are going to be motor. Okay? So now we've come behind the anterior nucleus. We still have some, what is VL? Ventrolateral. Now we've got some VPL, ventral posterior lateral, and luckily it comes with a ventral posterior medial. Maybe whoever named these was making more sense. All right? So now we've got VA and VL as this sort of anterior and somewhat dorsal tier. And that's motor. And then posterior to it, we're going to get VPL and VPM. And these are going to be somatosensory. So what have you just noticed? What's anterior to which? Motor is anterior to somatosensory. Think about cortex. What's the surface organization? Where is motor cortex? Precentral gyrus. Somatosensory cortex? Postcentral gyrus. Oh, look, the organization, the spatial organization is the same. Okay? So that, in fact, that's the landmarks that helps you do it. Emery.
So these slices, would it be like this if they were together? I guess I'm just confused about how it works.
So anterior, the very anterior part of the foramen of Monroe, a little further back, a little further back.
Okay, so they would be.
Yeah, so this is anterior here to posterior. They're basically stacked like that. So this slice here is this one pivoted, 15 degrees so you can see it. Okay? All right. So now we've moved further back. Where's the optic tract? Where is it? It's labeled for you. Well, it's all the way lateral, and in fact, it's reaching a gray matter structure, and that's, in fact, its thalamic target. That's the lateral geniculate nucleus. Okay? We'll see it at the end of the morning. There is the lateral geniculate. Okay? The optic tract is wrapped all the way around. What is this What is this domain right here? Yeah, that's midbrain. It has your three friends that let you know you're in the midbrain. The peduncle, the nigra, the red nucleus. What are the fibers that are here around the red nucleus? Where'd they come from? Cerebellum. Where are the rest of them headed? Thalamus. Where's the thalamus? It's right there. Okay? Here it is. This is how they got there. Okay? So they wrapped the red. We look at the red in this plane in the horizontal across the midbrain. Here, if you look at it in the coronal, it's a sphere. It's a sphere wrapped by those fibers, and as they go out the top end, they are heading to the thalamus. Now, they are angled actually out of this plane because they began posteriorly, and their trajectory is anterior. So they are, in fact, cerebellum fibers are going to what part of thalamus? What is that? What kind of information are you using that for? Motor. What is the motor part of thalamus? It's way back here. It's VAVL. So they're at that angle, running like this, not like that. Okay, so now we have more dorsomedial, lateral posterior, VPL, VPM. Okay, here we're going to have the hypothalamus. We'll worry about that later. Okay? So we've got those same structures coming back, okay? And as we finally come back, life gets really simple because the big lump at the back, that's probably 50% of the volume of the thalamus, gets only one name. And it's not a name based on location, so you can't worry about it based on it being in the wrong location. It is the pulvinar. Okay? And as we will see, the pulvinar is most intensely connected to the parietal lobe, and in fact, if I could spell it, and in fact to the multimodal association areas in it. Here is our lateral geniculate, right down here, the very tail end of the optic radiation coming into it, and next to it we have a medial geniculate. The lateral and medial geniculates, these two little feet that hang off the back end of the inferior surface of the thalamus. So, quick word on disease. What's wrong here? So I can make a diagnosis of a disease that this person had just by looking at the brain. So what do you see? First of all, where's the thalamus? But how many thalami can you see? One, because one of them is damaged by a massive hemorrhage. Okay, so here is our thalamus. Okay, are we anterior or posterior in the thalamus? What do you think? Here's the thalamus. So we're pretty posterior because you're way back at the level where you can actually see midbrain back here. And this is actually the geniculate. That's your lateral geniculate on that side. Okay? So you're in posterior thalamus. So this would be a little bit of dorsomedial. This would be, you'd be coming in towards pulvinar. Back here you'd probably have some, maybe the posterior boundary of VPL and VPM. You might even be behind it. And what's happened here? Blood. This is a thalamic hemorrhage. Major, by far the most common etiology for this is hypertension. So, poorly controlled hypertension, deep hemorrhages, particularly in thalamus, are very characteristic. And these tend to be fatal. They are not, they expand, they get into the ventricular system, they cause all sorts of problems. And as you can see in this case, this one was fatal. Why do you think something bad has happened here to the temporal lobe? Back to the very first day. What happens when you have a unilateral expanding mass? Yeah, brain herniates. It moves. So this person had transtentorial herniation on that side. That temporal lobe was completely necrotic. Let's take advantage here and look at the cortex. So notice as you come from the midline, I'm going to run my pen along the cortical ribbon. Where am I, roughly? What lobe? Probably still in the posterior portion of the frontal lobe. What gyrus would I be in? Imagine it visualized from the side. Well, this is probably what I would see as superior, this is what I would see as middle, and this is what I would see as inferior. This is my inferior temporal gyrus. Where am I now? Any ideas? Am I in the frontal lobe? Am I in the temporal lobe? I'm in the insula. That's your insula. Now I'm in my superior temporal gyrus, middle temporal gyrus, inferior temporal gyrus, parahippocampal gyrus, and then right here, as we'll see at the end of next week, the hippocampus. Completely continuous sheet all the way around. Okay? Why do I saw one little dead soul that I missed. Oh, yeah. So why is this sitting here looking like a separate little island? Notice that. Here's the cortical surface here. What's this? Is that some malformation? No, it's just the fact that you've cut across a folded, in-tucked piece from another gyrus, because it's a complex folded surface. So when we think about the thalamus, you should be able to draw this sort of football for yourself, okay? It's an oblate, it's at an angle in the brain, And then as you go from anterior to posterior, you should begin to basically think about what should I see and roughly what sort of function is it going to have. We already set up the sort of spatial dichotomy. Motor, as in cortex, is anterior to somatosensory. Auditory and visual are far more posterior. Okay? So as we go, we have the anterior nucleus that is the frontmost bulge. We have the long dorsomedial that sits along that superior and down the medial surface. We begin with ventral anterior and then ventral lateral. We come to ventral posterior and ventral posterior lateral, ventral posterior medial. Eventually we come back into the pulvinar, and we've got a couple of others, lateral, posterior, and then the centra median that I'll talk about at the very end of next week, of Friday. If we try and color code them and line them up with our cortical maps, and this is why I went through the cortex first, because now you can say, are they at all registered in space? And the answer is yes. And therefore learning them as pairs makes it somewhat easier. If you didn't have the cortex as an anchor, the thalamus seems completely random. It may seem random anyway, but it really isn't, because if you look at these maps and you say, well, gee, here's my difference between my blue, which is my motor, my ventral anterior and my blue here are motor, and those are out in front, in the posterior portion of the frontal lobe. What's more anterior here, the anterior nucleus is going to go to that whole separate structure. This is the limbics. And that dorsomedial, where does it go? Well, it's sort of medial. It goes to that whole prefrontal zone. So basically we can say this is prefrontal, this is limbic, this is motor, this is somatosensory, this green hatched area in the postcentral gyrus and the VPL, VPM. So that's motor, so that's posterior frontal. This is anterior parietal. As I said, the pulvinar comes to this vast associational area. This is the bulk of parietal, as well as some posterior temporal and some occipital. The medial geniculate is going to go to the superior temporal gyrus. And the lateral geniculate is going to go back out to Brodmann area 17 on the medial occipital. So we've got limbic, frontal, parietal, more posterior parietal, and occipital as you go from anterior to posterior along those slices of the thalamus. If you lay it out in this map and draw it in this sort of pseudo-compressed, this is sort of the equivalent of a maximum intensity projection of the entire brain, okay? So the thalamus doesn't sit in one plane like that. But if you flattened it into a plane and you flattened all of your cortical areas into a plane, how would they line up? And what you can see, okay, is that they don't cross each other, okay? They are able to draw from thalamus to cortex, no lines that cross over, essentially. Superior temporal. So it's very hard to see. It's this funny hatched area. It goes right in here. It's actually not really a surface thing. It's tucked in into the Sylvian fissure. That area is going to be, that's primary auditory cortex back there for the medial geniculate. The medial geniculate, we will see probably, I think now on Friday, not today, is going to be our auditory processing relay. All right. So, again, when we think about these, we had ones that were specific modalities. VPL and VPM are somatosensory. Lateral geniculate is vision. Medial geniculate is auditory. We have ones that are motor function. VA-VL will be connected to the premotor, supplemental motor, and to the motor cortex. Okay? So we've got those separations. We've got the ones that go to multimodal. The best example for us is going to be parietal. And we have the ones that go to the sort of cortical integrative functional activities. Dorsomedial, which goes to prefrontal, and then anterior nucleus to the cingulate. And then we'll talk about the intralaminars at the very end. So when we think about these, we now need to get our inputs into the cortex. So again, what type of cortices are these? The homotypic is mostly association. Okay? Heterotypic granular, motor or sensory? Sensory. And therefore heterotypic agranular, motor. So the inputs are going to go to different sorts of places in these. So for cortical inputs, they get inputs from other cortical areas. How do you go from a unimodal, from a first order to a second order somatosensory cortex? Well, that's a cortico-cortical connection. How do you bring sensory and visual information together? Those are cortical projections. So cortex projects to nearby fiber areas through association fibers, but you are in fact sharing information across the midline. How come? Do you live with two separate worlds, a left side and a right side? No. You live with one world, and in fact you are continually moving, so your vertical meridian is continually moving in physical space. You have to share information across the midline. And so every cortical area talks to its mirror image through fibers known as commissural fibers. They cross either in the anterior commissure for the temporal lobe or the corpus callosum for other structures. Okay? Cortex to cortex tends to go into supragranular layers, which you can translate into numbers as layers 1 to 3. Thalamus comes into layer 4, okay? Particularly for the relay nuclei, okay? For primary motor and primary sensory, that's where the input is, okay? We have diffuse projecting systems that we will get to at the very end, cholinergic, noradrenergic, serotonergic, dopaminergic. They are incredibly widespread. But what we've just done is basically say, if you think about a 6-layered cortex, where's my pen? If I've got a 6-layered cortex, 1, 2, 3, 4, 5, and 6. I've got one type of input that comes in here. I've got input from other cortical areas that are coming in here. And if I'm thinking about my output neurons, they're mostly here and here, with their dendrites going up to here and here. And these are my output structures, like this. So I've now got, as in the cerebellum, where we had distinct types of patterns of input, we've got distinct patterns of types of input into cerebral cortex. Okay? Oops. So the outputs, okay, cortex to cortex, the same way it got input, it sends output, okay? None of these are isolated under normal circumstances. The supragranular layer, what's in layer 3? What's layer 3? What type? External, pyramidal. In general, pyramidal are output neurons. Those are, in fact, commonly cortico-cortical projections. And then the deep structures, layers 5, are output into basal ganglia and into the motor system. And layer 6 is back to thalamus. And we're not going to talk about corticothalamic connections again, except you should know that they exist. And for lots of things, they are, in fact, really important. But life and our time together here in class is too short for me to cover them. So you've got different types of inputs. They are laminated. You have different types of outputs. They are laminated. Important basic concepts. So when we think about the thalamus, we've got, I don't quite know why data from rats is floating there. I was fiddling with slides, and that should be something in a different slide. It doesn't belong here. We've got the relay nuclei. Those relay nuclei typically go to layer 4. But sometimes they spread out, and the ones that are not relay nuclei tend to go much more widespread in cortex. Okay? The associational nuclei, they're not getting primary information. Where is their information coming? We've been sending our somatosensory information to VA, to VPL and VPM. We sent our visual information to the geniculate, lateral geniculate, our auditory information to the medial geniculate. What are we sending into the pulvinar? We're not sending cerebellar information there. Okay, cerebellar information is going to VAVL. It's part of the motor. What's going there? What's going to the anterior nucleus? So in fact, most of these are getting inputs from cortex. They are getting components of the data that they need to process, processing and feeding it back. So it's not just cortex to cortex that gives you your multimodal integration. It is in fact corticothalamic, thalamocortical connections that give you multimodal integration. And those tend to go as outputs mostly up to the upper layers. So let's see how far we can get. So when we think about thalamosensory, because I want to do the sensory first. We'll come back and do we'll get to motor and the other systems on Friday, I hope. We've got vision, our lateral geniculate, our medial geniculate. Remember, if I draw that oblate football, they are a lateral and a medial geniculate sitting off the back. Okay? That's because this is anterior, this is posterior, that makes this medial, lateral. This is a left thalamus. Okay? So lateral and medial geniculates. All of our somatic sensation, and in fact taste, because it is associated with your perception and sensory input on your tongue, even though they travel in completely different nerves, process in different places, end up together. And your vestibular information, the awareness of vestibular information, ends up in VPL and VPM. And this will make, again, a somatotopic map. And smell, remember, has no thalamic component. So where are the sensory organs? Where's the sensory cortex for this? So vision, Brodmann area, 17, primary visual cortex. Auditory, Brodmann area, 41. What we will know when we get to it, if maybe today, but more likely on Friday, posterior superior temporal gyrus, the flat horizontal part of it, not what you see on the surface, but what's tucked in underneath. Gets a name, it's Heschel's gyrus. Somatosensory information with the vestibular and taste for that stuff about your head is all in the postcentral gyrus, Brodmann area 312. Okay. Smell, we saw it, it's piriform cortex, it's Brodmann area 34. Okay. Just a quick, neat, this is where the rat data is supposed to, where the word that says most comes from rats. So it turns out that, in fact, when people look at cortical organization, it looks like rat piriform cortex is not a first-order cortex. And in fact, when we talk about it, the first-order cortical equivalent for olfaction, at least in a rat, and remember that rats do a whole lot more with smell than we do, is the olfactory bulb. So it really looks as though there is no thalamus, but the olfactory bulb, which actually is a layered gray matter structure, we don't think about it that way, but it is, okay, is in fact sort of our first-order sensory cortex for olfaction. Just because if you look at the connections of piriform cortex, it has connections not like a first-order somatosensory visual or auditory, but like a higher-order somatosensory cortex. And the argument is that the pre-piriform cortex, which is the primary olfactory, what was called primary olfactory, is actually an association cortex. So what you define as first-order versus second-order depends a lot on how you look at it. If you look just on the structural, you'd say it's the first cortical place. But if you say, well, it's defined not so much by where it sits, but by how the cells in it are wired, then it looks like it's a higher order cortex. So, let's think about the two geniculates, the two little feet that hang off the back. And I think we will make it through the lateral and then stop. So here's a brain dissected to show us visual pathways. So we've already talked about two visual pathways that are not really well represented here particularly. So what's the first that we talked about? Either one of two. So retinotectal And then the other is the retino, where am I? Retino pretectal, okay? We followed this information. Retino pretectal, the pretectal nucleus is going to drive what? Pupillary constriction. So the light shined in one eye, that information about vision, about light being present, not about any visual field, not about any perception, not about any localization in space. Too many photons for the diameter of my pupil, pupil constricts. Ambient light is dark, not enough photons for my retina to be in the good dynamic range. My pupil dilates to allow more light in. Okay? Very simple. Those are driven by photoreceptor, intrinsically photosensitive retinal ganglion cells that project from the eye back to the pretectal region. We also had that map of space that is really not about visual information, but about novel stimuli someplace in space that drive the tectum, superior colliculus, and drive your eyes to move to a new target in response to stimulus. Okay? But none of those are about your being aware of the world in a way that makes you conscious. So now we've got to get that information about perception to what? If you're going to be aware of it, it's got to get to thalamus and onto cortex. So what we've got here is a beautiful dissection. These are the stumps of the optic nerves. Here's the chiasm. Here are the optic tracts coming around, and they're coming back and tucking in underneath or superior to the midbrain. And what's superior to the midbrain? Thalamus. And what sits at the inferior posterior part of the thalamus? Those geniculates hanging off the back. And here we are coming, as they come around from the lateral aspect, right into the lateral geniculate. Okay? So this arc of the optic tract is coming right back here to the lateral geniculate. And in fact, as you go from the lateral geniculate, we will see the fibers arcing out and coming back to the medial aspect of the occipital lobe. What's on the medial aspect of the occipital lobe? Primary visual cortex. So we've done what we needed to do. We've taken visual information through the retina, brought it back, at the chiasm organized it into space, left and right, brought it to a thalamic nucleus, and brought it to the cortex. Job done. You now have gotten information to the cortex. Cortex can start doing visual processing. Where is the geniculate? Well, it's pretty posterior. It's pretty lateral. It's the lateral geniculate, okay, right here. It is shaped, the classic description is that it is shaped like Napoleon's hat, okay? It is a flat, layered structure that is peaked, okay? And depending on how you cut across it, you will be able to see some set of cross-sectional levels in different types of cells, okay? So it's this folded, peaked structure that is made up in any one of levels. Of layers, rather, of different types of neurons. Now, as the optic tract comes back on the right side of my brain, it is bringing information from which eye? Both eyes. About what side of space? Left side of space. So now you've got information about a point in space that is coming from your left eye and your right eye. And what happens at the geniculate is that they stay separate, so that your information is organized by eye in this direction, in layers, but by physical space in the sort of horizontal, so that if you go a trajectory down through the geniculate and record, you will hit cells that are stimulated by one eye, by the other eye, by the other eye, by one eye, by the other eye, the other eye. Okay? So that, but they will be from a point in space. If you move the source, you'd have to find a different spot in the geniculate to get the information from one eye and then the other and then the other and back and forth and so on. And the way it is packed is this. First thing is that there are 2 big cell layers and 4 little cell layers, magnocellular and parvocellular, okay? And 1, 4, and 6 are from the contralateral eye, and 2, I'll have to do it over here, 2, 3, and 5 are from the ipsilateral eye. All right? So information is still coded by which I saw it. Why would that be useful? Yeah, if you scramble those up, you lose depth perception. So in fact, that information is going to stay separate for multiple steps until you begin to make use of it to get depth.
And it's also still like left field, right field on the
No. So this is all, if this is a right geniculate, it is all about the left side of space. Okay? Because information segregated at the chiasm. Once you are behind the chiasm, it is only about space, not about field, not about eye. So when we look, you will see that as you come back into the right optic tract, it has information that came from the right eye and the left eye. They segregate out into layers, and this right lateral geniculate will go to right Brodmann area 17, okay? And we will have a complete map of the left side of the world. Okay? We will have it organized by space and organized by eye. Now, in fact, there's separations, and I think John will talk about this in the next block. There are different types of photoreceptors. They have different types of receptive fields. They are stimulated by different retinal ganglion cells, have different receptive fields depending on what pattern of photoreceptor anatomy or what spatial organization is activated. They have different characteristics in terms of their sensitivity to different colors, in terms of spatial resolution, in terms of sensitivity to contrast, and whether they fatigue over time. All of that information is going to basically be carried about every point in space. And each one of those is going to generate a separate sort of channel of information that you're going to bring back. And so, in fact, that separates out a little bit the difference between the parvocellular layers and the magnocellular layers. And just to make life complicated, it turns out if you're a physiologist, not an anatomist, if you're an anatomist, you see the big things. If you're a physiologist, it turns out that there are lots of other neurons in there that are in fact responsive to visual stimuli, that were always ignored. And they are what known as the conicellular. Those are the really little, smaller than the parvo. You have a question, Emery?
Yeah. Sorry, I'm just confused. You said the answer to John's question that it was separated by vision, or by visual field, but then you also said separated by.
Well, so they are the information. So you keep the information about a point in the visual field. So if this is my lateral geniculate, if I flatten my lateral geniculate out to a sheet, I have a map of space. Okay, this is the midline. This is superior. This is inferior. This is as far lateral as I go. Okay? In that sheet, if I look at it, it's actually a stack of sheets, right? It's got 2 magnocellular layers and 4 parvocellular layers, right? If I come down in any one place on that with my recording electrode, all of this column of cells are driven by one point in space. But they will be driven in layer 6, okay? 6 is part of 146, which is the contralateral eye. I will begin with the contralateral eye. As I come into layer 5, it will be driven for that point in space by the ipsilateral eye. Okay? Remember, most of your visual field is covered by both eyes. Okay? So that you have information in a layer comes from one or the other, one eye or the other. But as you come down that column, you've got information from both about that part of space. Okay. So then as we go from the cortex, This is thalamus. Where does it come into cortex? Sensory thalamus comes into layer. You got a 1 out of 6 chance of guessing right randomly. Comes into layer 4, okay? And it's going to come into layer 4 for the most part. The little tiny cells go up higher, actually. But you're going to come into layer 4. Now, in fact, you can see that where the gray matter comes in, where the thalamic projection comes in, and it is a line. Why is it a white? Why is it a black line? Because it is white matter. Okay, this is a myelin stained section. So you can see the white matter in the cortex, that line, and that is known as the line of Gennari. Okay? And it marks the primary visual cortex. And when we cut brains in lab next week, we will see the line of Gennari. Just a quick word, Gennari describes it in the occipital lobe. It's the easiest one to see. It turns out that there are ones in somatosensory cortex, and it's known as the band of Vic-Desir. This is Vic-Desir there. But all cortices, in fact, if you look hard enough, you can see a white matter terminal field in the layer 4. And that's known as the lines of Baillargeon. And he was the very first person to begin to try and say, what are the similarities as you look with, he actually looked at with a handheld lens. What are the similarities between cortical organization? What are the differences? He was the very first person to try and get at structural maps. So when we come into the visual cortex, and you'll hear a lot about this, and this is basically the organization that Hubel and Wiesel got the Nobel Prize for sorting out, is that as you come into cortex, you have ocular dominance columns. What does that mean? Well, it means that within this layer of cortex, this is a very small zone, you've got a stimulus that came in and was perceived in the left eye, and for the same place right next to it, the neurons will be driven by the right eye. So that means that the information about a point in space started in 2 separate retinas, came back in 2 optic nerves, came to the same side of the brain at the chiasm, travels back in the optic tract, goes to different layers in the lateral geniculate, because the lateral geniculate is layered by eye. From there, goes back in the optic radiation, those white matter bundles that go back to the occipital cortex, staying separate, and they actually know how to find a separate, slightly separate target right next to each other. So that as you move your electrode across the cortex and say, which eye drives it, it goes left, right, left, right, left, right. And as you move it in the other direction, you are driven by different directionality of the stimulus. And this is the fundamental visual cortical function. It is aware of which eye saw it. Based on where you are in the cortex, it's where it is in space. And the stimulus, as the first cortex sees it, is essentially angled lines. Yeah.
Do these ocular columns get bilateral projections from both lateral?
Nope. But each lateral geniculate, it gets information about one side of space from both eyes. Okay? So as we watch the optic fibers go back, they have this broad sweep. This is called the optic radiation. So we had an optic nerve, an optic chiasm, an optic tract, and an optic radiation. Okay? The optic radiation, you will notice, has this funny anterior fan. See how they spread out? It doesn't just go like that. It arches, and these run more anteriorly and turn around. That anterior portion sticks out into the temporal lobe, where it turns out to traverse into a different vascular bed, and it's known as Meyer's loop. One can see this, and one can map it, and you can actually say, what happens if people injure that? And in fact, what happens if people injure Meyer's loop is that they lose. What sort of visual field could you lose? Let's imagine you injure that loop on the left side. Would your view of the left side of the world be hurt? Your view of the right side would be hurt. And it turns out that Meyer's loop are fibers that cover mostly the upper quadrant. So what would you lose if you injure Meyer's loop on the left, what would you get? Left or right? Right, upper quadrant, enopsia. Both eyes would not see things that were in the right upper quadrant. So let's think about that. So let's look at
So it's
upper quadrant in space.
doesn't that also
No, we do things in terms of space, not retina. So it's upper space. We don't worry about where in the retina. Ophthalmologists worry about where in the retina. So let's look at what's going to happen if we put a lesion right here. Number 1. What are you going to get? So vision from this eye, normal. Vision from this eye. Okay. Let's put our next lesion out here. This is a really hard lesion to actually have physiologically, but it's this one. What are we getting here? It's little orange, little bridge. What are those? Well, they're fibers that began here and stayed on the same side. So they are fibers that are not crossing in the chiasm. So what are they fibers about? Contralateral space, right? And so from this eye, contralateral space is seen as vision to that, okay? And the other ones as well. So what will you get? What will you lose? You lose the center, all right? More common, and I'll show you an example in a moment, is when the lesion happens right here at the crossing fibers in the chiasm. And we talked about this. What do you get? Bitemporal hemianopsia. All right? You put a lesion back here, and you hit the optic tract as it goes to the geniculate. What do you lose? Contralateral for which eye? Both eyes. That's the homonymous hemianopsia. Both eyes have lost the same area of space, same half. Homonymous. Between the two eyes, it's the same. Hemianopsia. If you move back into the optic radiation, you get the same thing, unless you're at a point where you are separating out the fibers that are going to go for superior space and inferior space. And as we saw, those that go through Meyer's loop in the temporal lobe, where's my pen here? Meyer's loop in the temporal lobe gives you upper quadrant, and if you get the other fibers, you get an inferior quadrant. The visual association processing areas are all surrounding that cortex. So if this is our primary visual cortex, talk a little bit about the organization. Here in the right hemisphere, this is left space. This is the center of field going out to the periphery. So at the occipitalmost point, here is the center. And this is the periphery. The superior bank is inferior space, and the inferior bank is superior space. So we've got a map of that half. Finish with just a couple of quick pictures, and then we'll take. No, it's what's going to the superior bank is the way it's drawn. So superior bank is inferior space. What do you see? Where are we? Got a pair of eyes. What's hanging off the bottom? Well, it should be the optic nerve, right? And that's the optic nerve coming back to the chiasm. And on this side, it looks pretty normal. And on this side, definitely not normal. It's actually got glioma in it. This is actually a patient with neurofibromatosis type 1. They get hypothalamic and optic system gliomas. They present with unilateral blindness. Here, it depends on where they are, but this patient, it actually did not come back into the chiasm. It was all in the intraorbital segment. What do you see? Anything that doesn't belong? Well, there's this large lump here. And that's actually metastatic carcinoma. And what are these structures on either side? Yeah, those are the optic tracts just posterior to the chiasm. So the chiasm here has been completely destroyed by this. What would this present with? Bitemporal hemianopsia. Here we have a slice through the parieto-occipital region. The white matter of the optic radiation runs right here. So this person actually has a small infarct right there in the middle. What would you expect they would complain of? So in fact, what they would probably complain of is nothing. Because you've got a defect in the field. But because you move your eyes so much, unless it's huge, small defects you are pretty unaware of. Unless you ask them to be tested, in which case they're asked to look straight ahead, and on a field, visual stimuli are presented, and they're actually asked to tap a little button when they see it. It's the equivalent of doing this. When do you stop seeing it? And you can then get the contour of their visual field, and you'll see that it has a defect in it. But most people with small defects are completely unaware of it unless you test them. And here is a horizontal plane. What's the area that this is a posterior cerebral artery territory infarct. What is right back here? Well, think about the brain. Here's the brain. Oops, it's the edge of my screen. Here's the brain, okay? I've cut in the horizontal plane, sort of like this, and I'm looking at the posterior portion. What have I got on the medial surface right up against the midline on each side? Primary visual cortex would have sat right here and here. So if it's infarcted, the person will be functionally blind. Now, this person is dead. But if a person survives this, they will be cortically blind. They will not have visual processing information. Will they have a pupil reflex? Yes. Might they even be able to localize a stimulus in space? Yes. That goes with the colliculus, that retinotectal. They don't need cortex for that. Okay? All right. Let's stop there. I will shift the auditory stuff to Friday. We'll just talk afterwards, okay? The students who's in Jean's group, because Jean is out sick, so 3, 4. Why don't the two of you join John, and the other two join Arjun today? Jean, I unfortunately have a meeting, so I can't pick up her group, so join the two of them. Terrific.